Good afternoon. Fortunately, the sun has moved on, so you can see the screen, I hope. So I'm going to tell you in 10 minutes pharmacogenomics in clinical medicine. I'm going to start off with a definition. These are two definitions. One is on pharmacogenetics and one's on pharmacogenomics. And these were defined by the International Conference on Harmonization in November 2007. And really what pharmacogenetics is described by Vogel, who was a German pharmacologist in 1957, is a study of the variation in DNA and how it affects the way you respond to drugs. And pharmacogenomics was a term coined by Marshall in 1997. And according to this definition, includes RNA as well as DNA. But we tend to use the terms interchangeably. And in a way, all we're trying to do here is to really personalize drugs to the patient. So although this is from 1957, the term, in fact, pharmacogenetics is actually, or pharmacogenomics is much older. Goes back to the 6th century BC, from the time of Pythagoras. So if you've got a boring Sunday afternoon, you can look up Pythagoras. And he did a lot of writing, and one of the things he wrote in was that you should be far from fava beans consumption. And he held that belief so strongly that he was being chased by an army of rival soldiers, so they refused to cross a field of beans in ancient Italy, in Cretonia, and they caught him and killed him. And there are many theories as to why he didn't cross the field of beans. And one of those is that the fava beans look like testicles, and maybe it was related to that. But however, the real reason is that fava beans cause a condition called favism, some of you may have come across, which can cause red blood cell hemolysis, acute renal failure, etc. We now know that this is due to glucose 6-phosphate dehydrogenase deficiency, which is the most common human enzyme deficiency in the world. And the relevance to pharmacogenomics is that there are many drugs out there that we use which do cause hemolysis if a patient has a variation in glucose 6-phosphate dehydrogenase. And the first time I ever did a pharmacogenetic test was when I was an SHO at the Hammersmith Hospital in London. A lady came in with Vivax malaria, and I tested her for G6PD and then was able to give her Primaquine to treat her Vivax malaria. So if you look at pharmacogenomics and why we need personalized medicine as a whole, pharmacogenomics is one component of personalized medicine. Well, it's variability. We've known about variability for many centuries. William Osler, who's the father of medicine, really said it in the quote that you can see on the slide. And what we do at the moment is treat patients on the basis of one dose fits all. But given the variability, you can imagine one dose doesn't really fit all, because when you give a drug at the same dose to everybody, the variation exposure is enormous. And the exposure that you get in plasma equates to the exposure you get in tissues, and therefore you get variation in effects. And if you can generalize how drugs work, and how drugs are handled by the body, you can divide them into two particular categories, and I'll come back to that in a minute. But based on what's out there at the moment in terms of pharmacogenomics literature and in terms of drug prescribing information, this is a summary from the European Medicines Agency in terms of the drug label, the summary product characteristics. There have been 517 products licensed between 1995 to August 2014. And you can see about 15% of them have pharmacogenomics information. And I don't know, how many of you seen a summary of product characteristics recently? Have you looked at it? Please raise your hands if you have. Yes, very few. Summary of product characteristics has many different sections. And the section which is the most important is how you prescribe in terms of indication, what you prescribe it for. And what's in there should be mandatory. But only 3.5% of the pharmacogenomics labeled are in the therapeutics indication section. All of those are in the oncology area. Peter mentioned one of those, vemurafenib for metastatic malignant melanoma in BRAF positive patients. And the three in non-oncology areas. One is abacavir, which I'll mention in a minute. The other is in eliglustat, which is used in Gaucher's disease, and ivacaftor, which is a new drug licensed last year for treatment of cystic fibrosis. So rasburicase is a drug which does cause hemolysis in patients with G6PD deficiency. However, the label at the moment isn't in the indication section, but in the contraindication section. So if you are using rasburicase, you do need to test patients for G6PD deficiency. And this is a new drug, so G6PD is not relevant only to old drugs, but also new drugs which are being licensed right now. So generally, what you can do is to divide up the pharmacogenomic variations into two different areas. Pharmacokinetics is the way the body handles the drugs in terms of absorption, distribution, metabolism, and excretion. And pharmacodynamics is what the drug does to the body in terms of drug targets it works on, in terms of ion channels, receptors. And there's variation in all of those processes in both the pharmacokinetics and pharmacodynamics, and that can lead to complexity in terms of how we respond to the drugs, in terms of how we develop side effects. So here's an example of a pharmacokinetic. I'll go through two pharmacokinetic examples for you. Here's an example which is already in clinical practice, and if you're a rheumatologist or a gastroenterologist or a dermatologist, you may have used it when you prescribe patients azathioprine. This is deficiency of thiopurin methyltransferase, a very important enzyme which metabolizes these particular drugs. And if you have a deficiency of thiopurin methyltransferase, you get an accumulation of metabolites called thioguanines and so on. If you have too much thioguanine in your cells, that increases the risk of hemopoietic toxicity. And we know that 1 in 300 people have got homozygous deficiency in thiopurin methyltransferase, and you have to really reduce the dose of the drug that you're going to give them to prevent severe bone marrow suppression in those individuals. And that is used at the moment. And in kids, when we give 6-mercaptopurine for acute lymphoblastic leukemia, the dose is individualized on the basis of that. Here's another example which may be coming to primary care in the near future. This is with warfarin, very widely used drugs, but dose varies quite a lot from 0.5 to 20 milligrams a day. We published a trial in the New England Journal of Medicine which showed that genotype-guided dosing using 2 genes was better than standard dosing that we use currently in the NHS, with a 7% difference, but the difference was actually greater in those individuals who had the rare variants. And we've also shown this to be cost-effective. The cost effectiveness shows it's £5,000 per quality adjusted life year, which is well below the NICE threshold. So what we're doing at the moment is implementing that in 3 pilot sites in the Northwest to see how it works in a primary care setting before we take it out into the wider NHS. Now, in terms of the pharmacodynamic, the biggest area where there's been advances is in terms of serious adverse drug reactions, such as toxic epidermal necrolysis or hepatitis, etc. And the most important example is with a drug called abacavir, which is in HIV, which causes hypersensitivity reactions in about 5 to 7% of individuals. And work which was carried out in Australia, in the United States, and in the UK was able to show an association with this particular HLA allele. And we, at the same time, undertook a cost-effective analysis at the same time as we actually identified this in the UK population. And in this cost-effective analysis, we were able to show that by genotyping individuals for this particular allele, we would be able to save money for the NHS. So this paper was published in 2004. In 2006, every HIV clinic in the UK was using genetic testing for abacavir before they put patients on abacavir. And the net effect of that has been dramatic. Before testing for HLA-B57, the incidence was 7%, right down to 0, less than 1%. I haven't seen a case of abacavir hypersensitivity for the last 5 years because of genetic testing. So that tells you genetic testing can actually work in terms of improving drug therapy. So we've got so many variants, we can't really do randomized controlled trials in every area. And one possibility is to actually make that genomic testing available to you in your electronic medical record when you see the patient. Now, we don't have the infrastructure in many areas at the moment. In primary care you do, but in secondary care, very, very few trusts at the moment have electronic medical records, but that's coming through. But in the United States, what they're doing is actually preemptively genotyping individuals. And in this particular experiment that they're doing at Vanderbilt. They preemptively genotype individuals. That particular genotype is available in the electronic medical record. When a patient comes to their doctor saying they want to be prescribed drug X, a pop-up comes up saying this patient has a genetic variant in that, and then the doctor can then decide whether they want to change the dose or give them an alternative drug based on that preemptive genetic testing. And the advantages are many, and one of those advantages is actually cost saving as well. If they were to do this reactively, in terms of genetically test, it would actually cost more than the preemptive genotyping that they undertook. And you may say, well, is it really worth doing that? Well, for our patients. But so let me show you some data, and this is a slide provided by Dan Roden, who's a professor of cardiology in Vanderbilt. And these are 5 different phenotypes at the bottom, which are sort of known, very well known from the literature to affect the way those drugs work, so clopidogrel, warfarin, etc. And the red is what's a high-risk variant, and the blue is an actionable variant. And when you put it all together, those 5 different variants, 91% of you in this room will have at least one of those variants. And if you were to require one of those drugs, would you rather that you were given the right dose rather than the dose that we use at the moment? So I'll leave that for you to ponder. So just to finish off, pharmacogenomics is a part of personalized medicine. It is one aspect of a complex and evolving field. Its contribution to disease process optimization of therapy will vary according to which area you're looking at, etc. But it needs to be considered together with other technologies for personalization. Implementation is difficult because genetic testing is still regarded as a disruptive innovation. Clinical methods of stratification, all of us use Chad's vascular score for seeing whether patients require treatment with warfarin for atrial fibrillation. But we can use those, but there's no clinical trial of those, but we accept those because there's no testing required. In implementing these kind of biomarkers into clinical practice is going to be difficult, and it is going to require a lot of effort from all of us. So thank you very much for your attention.